Hi, and welcome to Worldwide Clinical Trials' latest webinar. Thank you for joining us. I'll now introduce you to our speakers. Enjoy!
It's a wonderful time to be in clinical research, very advanced technologies, very nuanced patient targets. Never has it been more important to have an integrated earlier phase discovery plan. So our question for today, is it normal volunteers? Is it patients? Or is it hybrid studies? Or as once has been said, should we build the bridge slowly to cross it quickly? I'm Michael Murphy, Chief Medical and Scientific Officer for Worldwide Clinical Trials, and I'm joined by my colleague today, Sherilyn.
Hi, this is Sherilyn Adcock. I'm the Executive Vice President in the Medical and Scientific Affairs Division. I support early phase, pharmacokinetics, and bioanalytical services.
Okay.
Nice to be here.
Our first theme for today is the formidable task of adjudicating the perspective of multiple stakeholders in phase one, who frequently have conflicting but equally important perspectives in terms of the type of data that need to be acquired. So our topic is, how do we satisfy medical, scientific, and business mandates with the constrained resources that are so common in pharmaceutical R&D today? Our next slide gives you a sense of what that looks like. So in an integrated early clinical development program, the scientific mandate, of course, rules. Never in clinical development would preclinical data have more impact on the nature of the experimental design. We're asking questions such as, are the pharmacological and indeed pharmaceutical properties acceptable? Does the compound distribute to target space or tissue? Does it engage target in humans, thereby validating the preclinical pharmacology that everyone has invested so much time in? Can we estimate what a therapeutic index in man could be? And here it is. Can the target engagement data be linked to the intended clinical impact? These are our basic questions, but it goes beyond that. In the next slide, we need to adjudicate the reality of the environment in which this occurs, the business mandate. Is it a repurposing product? Is it a novel chemical or biological entity? Or is it increasingly an advanced therapy medicinal product such as cell, tissue, or gene therapy? Does the company have a single asset, or is it a platform technology with a lineage of possibilities beyond the one that's currently under review? Should we recommend an established pathway, that is, an established R&D pathway, where the value of the pathway is in fact that it's predictable? Or, in contrast, given the company's interest, should we first evaluate what I call targets, then pick the indication? The value in that program would be what I call discovery and development. In essence, bringing the discovery research into the clinical development space. One can see the conflicting but equally interesting possibilities. And finally, in every single case, I say we must live within your means. And that means we need to develop this product to what I call company inflection points, points in the program where the data inform the next step and it occurs in a timely, predictable, and thorough fashion. So satisfying medical, scientific, and business mandate is very much the assignment that we need to address in early phase clinical research. So the biggest issue, and the one that's ubiquitous across therapeutic areas, phases of research, technologies, is what I call DER, dose exposure response. You know, the general sentiment out there, by the way, in our experience, is more doses, but fewer patients per dose, and the emergence of what I call trial complexity. Sherilyn will share that experience with you because she's in the trenches.
Very good. Thank you, Mike. There are certainly many questions to ask in early clinical development, and this requires a well-orchestrated plan. A comprehensive investigator brochure that clearly tells the story of preclinical data is the basis for moving forward. In addition, the transfer of bioanalytical methodology from preclinical animal models must be used to develop and validate bioanalytical assays in human matrices for plasma, urine, cerebral spinal fluid, and other bodily fluids. The bioanalytical assays must be well developed and robust, and the clinical pharmacology unit drawing those PK samples and PD samples must fully understand how those samples are to be collected, processed, and stored in order for results to be useful to establish pharmacodynamic behavior of these investigational products. For the orphan disease patient, in particular, these populations, it's important to consider any limitations and requirements that might impede or discourage patient participation. Ensure that, for example, that patients and possibly caregivers as well are provided for with the appropriate and comfortable accommodations in the clinical pharmacology units, and that the number of procedures such as blood draws and particularly other invasive procedures do not result in unnecessary stress on the patient. There may be a tendency and an interest to include a number of intensive assessments, but to ensure that there is scientific basis to support these test procedures is very important. In early phase development, the scientific and clinical evaluations may require additions or adjustments based on the data received from previous dose level cohorts, and thus the scientific evaluation support of these are ongoing process, subject to adjustment and need for adaptive designs. Clearly, there's a value in pushing the envelope within the constraints of safety, patient acceptance, and budget. The more data, the better. The clinical pharmacology unit is critical to managing all the moving parts in this early development programs. These units must be well staffed and well equipped for managing the logistics of multiple procedures in a short time span, ideally including an on-site pharmacy for formulation services, clinical diagnostics, safety laboratories, and full-time investigators and medical staff that are really dedicated to the clinical research aspects of each study. Special assessments will be discussed in more detail in this presentation. And however, it's important that special assessments specifically related to orphan diseases under evaluation be on site at the clinical pharmacology unit or in close proximity so as not to disrupt the flow of procedures or unduly inconvenience the patients.
To really put some emphasis on what you said there for orphan diseases, I can't tell you how enabling that microanalysis and sampling technique that you have there has been for many of our orphan indications. It just transforms the whole nature of the trial, particularly for very young children, where
Yeah.
it's impossible to do the typical venipuncture approach that we might do in adults.
Okay.
So I really wanted to
Yeah.
echo that. It just enables a whole different dimension for trial design.
Yeah, and earlier we discussed the timing of these method adaptations from animal models into humans. New chemical entities may require custom bioanalytical assays with potentially multiple analyte measurements. These bioanalytical assays should be fit for purpose with the appropriate assay ranges identified early on to allow for the evaluable data at the lowest and above the highest expected dose to avoid the gaps in questionable data with respect to the pharmacokinetic profile. Now, the plasma, serum, and whole blood, and even urine are commonly evaluated from a pharmacokinetic perspective. However, orphan disease states may require rare matrices that, especially to elucidate biomarkers. As these procedures are typically invasive and require special techniques, again, as in the case of cerebral spinal fluid collections or dermal biopsies, it's important to keep in mind how orphan disease patients will tolerate these procedures and carefully consider the intensity and frequency. Choose your time points wisely so that these procedures can be accomplished. You want to correlate possibly with the expected pharmacokinetic parameters, such as the maximum concentration time and the distribution profiles as these are available. In addition, the volume of blood or other bodily fluids needed to perform the bioanalysis is critical to know before any sample is ever taken. Assays should be developed to minimize the amount of blood required for these patient populations in particular at each PK draw, and also consideration of the total volume of blood required for the study, which may also take into account blood volume needed for clinical safety laboratory assessments. Now, other considerations associated with bioanalysis include low-level quantitation analysis. This is not always necessary, but when it is, it's highly desirable to contract with a bioanalytical lab with the instrumentation and technical expertise to develop assays even in the picogram range. In addition, the bioanalytical laboratory is an integral part of the timeline for these early development studies. So prior to dose escalation occurring in a clinical setting, it is often necessary to have the bioanalytical PK data as well as clinical safety data prior to start proceeding to the next dose level to be initiated. This requires close coordination between the clinical pharmacology unit and the bioanalytical lab and allows for the seamless transfer of bioanalytical samples once the last sample to be analyzed is drawn, allowing for timely receipt, accessioning, and subsequent analysis of those bioanalytical samples by the lab, and followed immediately by pharmacokinetic analysis and report findings to the dose escalation decision committees. Ideally, you'd have a very strong, experienced project manager that would oversee all these timelines for all these different departments, from clinical through bioanalytical, data management, biostatistics, PK analysis, and final report. Now, one of the things that we mentioned in this particular slide has to do with mass balance studies. These can provide valuable information with respect to drug absorption, metabolism, and elimination. These studies are conducted in a small cohort of generally of normal healthy volunteers. Metabolic profiling associated with these studies is useful in elucidating the number of metabolites and their activity or lack thereof. The bioanalytical lab can use this data, if available, to develop multi-analyte assays as needed. If there is expectation that certain patient populations or certain conditions have significant influence on the absorption, metabolism, or elimination of a compound, which may be the case in some orphan diseases, then conducting these studies in patient populations may be merited. In one case in particular that comes to mind, we did a mass balance study in end-stage renal disease patients that were on dialysis. One of the more recent innovations, as Mike mentioned earlier, and is especially useful in children and patients with limited venous access, is the use of microanalysis, where small amounts of blood, usually less than 1/2 ml, are collected in either capillary tubes or even using the dried blood spot technique. Before these procedures can be employed, however, it's important that the bioanalytical laboratory have a validated method associated with this technique.
Well, Sherilyn, fabulous presentation emphasizing the incredible importance of the dose exposure response relationships in early phase research, and particularly in the orphan disease space. But somewhat equally important is this particular slide, which I've always been fascinated from the time you first introduced me to the concept. I simplistically call it a drug in a bottle approach. And its importance is related to the fact that it allows sponsors to go in with a totally flexible picture regarding the possible dose range of interest without committing themselves to any particular dose. So, Sherilyn, if you could kind of walk us through this one, because I really like this one from a drug development perspective.
I'll be happy to, Mike. And of course, this is near and dear to my heart as a pharmacist, and particularly a pharmacist involved in formulation work. Really enjoy including this and having helped to establish these procedures at our facility in San Antonio, Texas. Clearly, a knowledgeable pharmacy staff with early phase clinical research experience can play an invaluable role in the successful outcome in these early development programs. The pharmacy staff should be involved very early in the process, from initiating the study in the pharmacology unit, from the preparing for drug receipt, making sure you have the right storage conditions available. This is absolutely critical to having a well-controlled and well-behaved investigational product, from initial assistance with formulation development, capability for dose adjustment between each cohort, and dose administration techniques. The pharmacists and the pharmacy technicians and our quality control staff in the pharmacy can help to ensure that there's a quality investigational product and help move the timeline along without significant delays and costs associated with contract manufacturing, especially when unexpected dose adjustments are required. Now, the above photograph shows the powder in bottle procedure where individual doses of investigational product were weighed into amber glass bottles. In this particular study, batches of 125 bottles were prepared with a specific milligram quantity, plus or minus 3% range, and later, just prior to dose, designated bottles were assigned to specific patients per randomization, reconstituted, and administered in a multiple ascending dose study cohort. Matching placebo bottles were filled with an equal volume of diluent only and administered to patients randomized to the placebo dose. The powder and bottle batch for the next dose escalation cohort was prepared in the same manner only after Safety Review Committee approved the next dose level and provided written documentation to the pharmacy staff. Similar procedures are available for just-in-time capsule filling, solution and suspension preparation, and of course mixing hot and cold investigational product for radiolabel AME studies. Now, the minimum requirements for these procedures include a dedicated space with appropriate air filtration, ability to store investigational product under the required storage conditions, both before and after manufacture, continuous security and environmental temperature, humidity, lighting monitoring in the storage facility. And oftentimes in the dosing areas as well, where a drug may be light sensitive, we may draw PK samples under amber lighting conditions. Now, adherence to FDA CGP guidance for phase 1 preparation of investigational drugs is a must. These include well-defined written procedures, adequately controlled equipment and manufacturing environment, and accurate, detailed, and clearly consistently recorded data related to the entire manufacturing process.
So, Sherilyn, we've often had this discussion. My data are fairly old, but I have the impression that this capability can result in a significant savings to a typical R&D group who now don't need to prepare individual dosage strengths for dose ranging purposes, particularly when they really have no idea where the effective or reasonable dose range might be. Do you have any insights in that regard?
Yes, Mike, that's a very good question. We have had one large pharma company that indicated to us that by making the individual batches and it being able to adjust for the single ascending dose study saved them approximately $250,000 in cost for each study they conducted. And it's not just the monetary costs, it's the costs associated, and that doesn't even include costs associated with the time delays which might occur first to even get into a contract manufacturing lab, or the time to get the product shipped and to get it assessed, the time to develop stability. If long-term stability or even short-term stability is needed, we can make these products just in time and provide a quality product for dose administration.
Sherilyn, here's another aspect to this now that's quite fascinating. Is it in principle possible for your pharmacy service to have this available for a typical outpatient study, let's say in a given orphan disease? Could we, in fact, use this approach or not to conduct, say, a multi-center study in children with a given orphan indication?
Certainly, it's something we can take a look at. We look at each protocol, and there's certainly specifics and nuances to each protocol. I think our team is very adept at coming up with creative solutions for situations such as this. And even last year alone, we made over 7,000 capsules for a phase 1B multi-center patient study.
Fantastic. Fantastic. Now, I know that specialized measures are also key, Sherilyn, and you have a considerable amount of experience and capability at the unit. And I think on our next slide, we can illustrate what those are.
Sure. There are actually a number of special assessments that can be of interest during the early phase development of an investigational product that's with or without an orphan indication. The list on this slide is certainly not meant to be inclusive, although the primary objective of an early phase studies is safety, tolerability, and pharmacokinetics for the most part. Clearly, including special assessments relative to the intended indication, can be insightful and bring value to the compound under development. For compounds with central nervous system indications, there are well over 40 validated instruments to consider. CRO that has an established reputation within clinical assessment technologies can be an invaluable resource in helping to choose the appropriate assessments to best fit your compound's expected profile. Now, imaging techniques may also be available on site or in a nearby radiology and imaging center. It's very helpful to work with a clinical pharmacology unit that has that established relationship with the imaging center and that also has the interest and willingness to conduct clinical research at their site. Whether the studies are in healthy volunteers or in patients, a full suite of non-invasive cardiovascular assessments is needed to evaluate cardiovascular issues that could arise with an investigational product. Clinical pharmacology units must be able to offer real-time evaluation of ECGs and telemetry to managing data capture for subsequent evaluation if deemed necessary. It's better to have that data and to store it as opposed to not have it and wish you had it at a later point. The clinical pharmacology unit usually works closely with a cardiac core lab vendor that can offer expertise in evaluation of cardiac effect, like QT changes in particular, with precision that may allow for a waiver for conducting a full TQT study at a later date. Now, most recently, the FDA has issued a draft guidance related to the assessment of pressure effects of drugs. The guidance states that elevated blood pressure is known to increase the risk of stroke, heart attack, and death. Therefore, the effect of an investigational drug on blood pressure can be an important consideration in the benefit risk assessment. We've observed the use of ambulatory blood pressure monitoring more and more in the early phase development studies or in early phase 2 studies, where you obtain a 24-hour baseline reading prior to administering the first dose, and then again after completion of the multiple dose phase of the study. Clearly, early single dose escalation studies may utilize multiple blood pressure measurements to evaluate any potential pressure effects, and these are evaluated in generally real time. Now, continuous and/or intermittent cerebral spinal fluid collections are an invasive procedure that cause limitations to study participants' ability to be ambulatory for a period of time, and they do pose a higher risk to the participant. Careful consideration of the expected value of this data is important. Be sure it's something you need. Comparison of the pharmacokinetic data from the blood and from cerebral spinal fluid can certainly provide a valuable information about the drug's ability to cross the blood-brain barrier and to what extent it crosses. Now, clinical pharmacology units that perform these procedures have highly skilled medical staff and physicians, including anesthesiologists, that perform the procedures and manage participant expectations and comfort needs. CSF collections may also be used to evaluate disease-related biomarkers.
Sherilyn, a question always comes up. What's the minimal interval between successive CSF acquisition? How close can you make it in a typical normal volunteer?
Well, it really depends on whether you're looking at single individual venipunctures, so single individual spinal taps, or you're looking at a continuous collection over a 36-hour period. We have a technique that is utilized in our clinic in San Antonio where we collect very small amounts of cerebral spinal fluid intermittently, even as often as every 10 minutes, into micro cryovials, and we can collect that over a 36-hour period.
Wow, fantastic technology. So many hypotheses. You know, the problem we get into, though, in my experience, is I often feel we're adding procedures and hypotheses like barnacles on the bottom of a ship. And we're introducing considerable complexity into some of these early phase programs. So balancing the question against the procedures always is really a fine art. And this next slide kind of illustrates what people have been saying for some time now. The original article was in 2011, it's over a decade ago. If anything, this has become accentuated over the last 10 years. And what it's looking at are the number of protocol mandated procedures looking at two different epochs. but also mapping it against total work burden by the staff. And it does so by therapeutic area. As you can see, there's been a dramatic upturn in the total work burden on the right-hand side of that slide over the last decade. Really incredible. So the ability to manage staff, manage processes in a first in human study is really key in order to permit us to ask these interesting questions. Now, when I looked at this originally, I thought for sure that central nervous system disorders would be at the top of the list. But surprisingly, anti-infectives and cardiovascular, as well as a little interesting readout there on immunomodulation, really reign supreme. So interesting questions, complex procedures. At the same time, a unit needs to have terrific facility with training and managing staff and intercalating all these different measures into a typical protocol. That kind of brings us up to our next topic for today, which deals with the question of, you know, is it, should we do healthy volunteers? I mean, is that really the traditional way to go? Should we do only patients? I would say every week I have that discussion. Or should we do both through a hybrid design? Or as Sherilyn, I know you refer to it, let's do just one more cohort, right? So this is part of the idea of, do I continue a discovery process in clinical development, or do I engage in a very traditional structured program? So to illustrate possibilities, what we have here is an interesting challenge. So let's say that I have let's call it an immune modulating agent of some sort. I need to make a decision, whether it's normals or patients or some other permutations. And what we're going to do here is just walk through some of the advantages and disadvantages associated with both. So my colleague Sherilyn is going to take the normal healthy volunteers and I'll do patients. So take it away, Sherilyn, please.
Very good. I will be taking the left side of the slide and then passing over to Mike, although I will steal some of Mike's thunder by sneaking in a patient cohort or two with my healthy volunteers. Years ago, early development included very distinct and separate protocols for single ascending dose studies, followed by multiple ascending dose studies, followed by food effect for oral medications, and then other separate studies for drug-drug interactions in special populations, such as patients with renal and hepatic impairment. It seemed that biostatisticians were not necessarily comfortable with too many variables in one protocol. Today, it is not uncommon to see single ascending dose, multiple ascending dose, food effect, drug-drug interaction, and even maybe a cohort of special patient populations or two for a proposed indication wrapped up into one protocol, thus the hybrid design. Now, healthy volunteer studies have been the mainstay for early development, unless a drug clearly could not be administered into healthy individuals, which is the case with cytotoxic compounds. Healthy volunteer studies help to inform on multiple indications. Recruitment's much easier and expeditious with healthy volunteers, and in general, PK safety and maximum tolerated dose in healthy volunteers translates well into patient populations and helps to identify that starting dose level in patient populations. Possible pharmacodynamic markers may be identified in healthy volunteers, but collection on a broad range of PD markers for multiple indications is costly and likely not feasible for what could be questionable or no data. Healthy volunteer studies take the approach that there is no disease-specific biodisposition or disease-specific safety concern. This may be a particular consequence when it relates to orphan disease states. Now, starting with healthy volunteers initially, and then once the dose level of interest is established, moving to a cohort of patients with the intended indication can be a valuable approach, resulting in the ability to compare across both healthy and patient populations and to save limited and scarce patients for meaningful dose levels once safety and tolerability in healthy volunteers is known. Mike?
Yeah. Now, on the other side, people will approach me and say, you don't understand my business mandate. If I don't show something interesting in a clinical indication and soon, I'm going to have difficulty raising funds in the current environment. I need to accelerate the program. Isn't it true that if I targeted only a select indication, like in this example, rheumatoid or plaque psoriasis, isn't it true that I would accrue significant advantages? And what we always say is it is true, but around here, there is not a protocol or program design that one could advance that does not have advantages and disadvantages associated with it, and such is the case here. So on the upside, I can certainly get PK data in a SAD/MAD type structure. I can certainly look at safety information at maximally well-tolerated dose, a number of different definitions for that, by the way. I can use specific disease-related biomarkers, not simply pharmacodynamic biomarkers related to the compound's pharmacology, but disease-specific biomarkers that suggest a putative clinical effect could occur with larger sample sizes and longer durations of exposure. Therefore, I can look at efficacy, variously defined, provided I have an enabling ADME-TOX package, an enabling package of safety pharmacology, pharmacokinetics, biodisposition. And indeed, if I did that, I would have PK safety, maybe target engagement in the relevant population, not in the normal volunteer. Well, the problem is, of course, that that does not generalize. Unlike the program on the left, I can't assume that it's equally informative for all the other possible indications. Look at that graphic on the left. I've got rheumatoid at the top and multiple sclerosis at the bottom. Now, as useful as an approach in normal volunteers is, the usual questions we get would sound something like, I need to demonstrate some evidence of possible clinical utility as quickly as I can in a relevant patient population. Why am I not doing this study in patients rather than normal volunteers? And as most things are in life, there are no program or protocol design considerations that are not associated with advantages and disadvantages. And that goes across every therapeutic area and every type of technology. Well, the advantages with patients for a first in human study are considerable. I can certainly get PK in a SAD/MAD type of program. I can determine the maximally well-tolerated dose in the relevant population. Very standard approaches for doing that. I can obviously look at pharmacodynamic biomarkers, those biomarkers which are a reflection of a compound's pharmacology. Or I can look at disease-related biomarkers, which give me some inkling that says, with a larger sample size, maybe a longer duration of exposure, these are proxies, perhaps, of an eventual clinically important effect. So I can even, depending on how long I do it, I can even measure aspects of clinical efficacy if you give us enabling ADME-TOX data um, beyond the customary 2-species 28-day toxicology studies that many sponsors will supply. So a lot of advantages. And in fact, I'll get PK safety, maybe biomarker data in the exact population where I ultimately want to develop the drug, bypassing the normal volunteer program. Well, the disadvantages could be formidable. Number one, this stratagem does not generalize. You can see on the left-hand side of that slide, I have rheumatoid arthritis at the top and multiple sclerosis at the bottom, worlds apart in terms of clinically related issues. So yes, it informs an indication, but it does not inform adequately all indications. If it's a patient population, I have significant recruitment problems. That's because a prototypical first-in-human study with patients uses what we call a highly leveraged patient sample, patients that are somewhat idiosyncratic to those that we might ultimately treat, patients who have been deliberately selected to maximize signal detection so that it informs the design of the remainder of the program. So recruitment can be an issue. And in a rare disease space, perhaps a considerable one, well, because patients are ill, they take many other medications. So the whole mosaic of concomitant medications comes into play, which of course needs to be mapped into the drug-drug interaction data that one might have from your preclinical program. Finally, we've got to manage these patients. They generally are sick. They have a number of different specific needs. So it isn't every research unit that's capable of doing that while they're simultaneously obtaining the other information. And finally, we occasionally encounter situations, I must say it's not frequent, where there's an ethical and regulatory constraint. If one thought about it for a minute, it's the rare indication that would be substantially benefited by a short duration of exposure with a limited sample size. It doesn't actually speak to the specific patient needs of those individuals that I have in the study. So conflicting but equally interesting perspectives here. Well, like most things in life, there is a way to adjudicate these differences. The concept of program and protocol design at its foundation is the art of adjudicating multiple conflicting perspectives in early phase research. And the next slide gives us an illustration of how that could look. So on the left-hand side, we have a very typical first-in-human study. On the right-hand side, we have a somewhat unusual subsequent program. So Sherilyn, if you could walk through the left-hand side, this is an example of what you might do, and then I'll take those on the right.
So on the left-hand side, we're looking at a single ascending dose study in normal healthy volunteers, placebo-controlled, 8 on active treatment, 2 on placebo. And as the dose escalates, we have a 7-day waiting or evaluation period to evaluate the dose, evaluate safety, possibly even PK data to make a determination of what the next dose level might be. And we can continue this. In this particular case, we show 3 cohorts. I've had as many as 13 cohort in these studies. Generally, I would say probably 3 to 5 cohorts is the norm, again, in normal healthy volunteers.
Mm-hmm.
Mike?
And yes, and indeed, now everyone, those cohort sizes are quite arbitrary. They're interesting articles which have been published that survey industry conventions on cohort composition, let's call it that way, that clearly deal with starting dose, dosing increments, estimation of what the maximally well-tolerated dose would be. There's quite a body of evidence, which is a big advantage that I think is not appreciated. That design on the left, including any number of permutations, is informed by a huge literature that enables a very varying permutations, methods of analysis and interpretation. Now, as we go to the right, we start to get a little more interesting here. This is what I call a proof of mechanism or a target engagement study. You notice we've shifted to patients. Arbitrarily and quite arbitrarily, we have an imbalanced randomization of drug versus placebo. Why is that? Because I need to know what the drug is doing more than what the placebo is doing, so to speak, and we can collapse the placebo across those particular cohorts. Notice how we're only using the maximally well-tolerated dose. That is under the assumption that the dose exposure response relationship is monotonic, which means it's either linear or it asymptotes with increasing dose. For certain novel pharmacology, that is not true. So it would not be appropriate to assume that the maximally well tolerated dose is, in fact, the most efficacious dose, and that would complicate this relationship. But then what we might do in this hybrid design that we're alluding to is drop that information the way that I express it into a protocol, which technically is a proof of concept and in some respects might even be a potentially pivotal study depending on the indication and the technology. For an example, various permutations of adaptive trial methodology. So as we go from left to right, we start out with normals because it's so informative. Does it get in? Does the product hang around long enough to have a handshake that's clinically interesting? What are the big ticket safety issues we might encounter? That's invaluable information before we enroll sick patients into the next sequence here. Then we ask the question, is it engaging target in a disease state at a dose level that is, I'd say, maximally well tolerated? And then finally, can we parlay that? Can we translate that into the next study, which frankly, in orphan disease states, might be the only study, depending on the disease, using various innovative experimental designs, which are increasingly sanctioned by the community and by regulatory authorities. So a hybrid approach where you basically do both. So having said that, let's kind of wrap up our conclusions here for today. So at the top, we have what I would call more of a discovery in development rather than a development motif. This is a highly integrated early phase orphan drug development program. Preclinical data informs the clinical design and the clinical program at no greater point in development. This is where all the difficult task of designing and promoting these compounds really pays off. It informs how we think about the design, how we monitor efficacy or potential efficacy, how we perform surveillance mechanisms for safety. It's as much hypothesis generation as it is hypothesis testing. But it's our belief with innovative programs such as this, there's the maximum generation of information with minimal levels of expenditures when frankly, virtually everything is unknown. And Sherilyn, you could comment more about the integration that we often speak to.
As far as the continuing to move a product forward, these early phase development studies are absolutely critical. Having a comprehensive suite of services, including pharmacy, bioanalysis, clinical safety lab assessments, as well as any special evaluation considerations, is really key to moving these and getting into an area to demonstrate some target engagement. We have to have in phase 1 studies flexibility of design, and certainly a range of special procedures and assessments that are necessary. For the sponsor, there's usually 3 critical factors that relate to clinical trials in every phase of development. Data is one. This means high quality data and lots of it. Timely execution, no recruitment delays, whether this is in normal healthy volunteers or the expectation of it surrounds patient recruitment. How fast can you get me your data so I can make the next move to meet the timelines imposed by investors or whomever? The flexibility to make those changes and to adjust to those timelines is needed, and expedited back-end services such as PK, DM stats, and reports of based on ongoing findings. And clearly, in addition to data and time cost, the best value does not always translate to the lowest cost. Cost clearly is important to some, more important to some stakeholders than others. So oftentimes as we have these discussions with sponsors, we ask, which of these are most important to you? Time, data, or cost? And clearly, we will oftentimes jokingly say, pick 2. Clearly, all of these factors are important in clinical research, and the ranking of these, the importance of these factors depends on the stakeholder. From a scientific perspective, the clinical pharmacologist is the key stakeholder in early phase development, and quality data and timely execution are the 2 top considerations.
Well, as a clinical trialist, it's a wonderful opportunity to showcase the art of trial design. Because again, as we've emphasized today, early phase clinical research, and by that I do not only mean first in human, perhaps it's first in human, it's target engagement, proof of principle, and so on. That's when the value of the product is truly established. So the art of clinical research is really about balancing multiple conflicting interests of patients, families, investors, discovery, development staff. All of these are stakeholders. And by doing this in an effective way, you produce an integrated demonstration of product value in the context of evaluating product novelty. So we hope that you've enjoyed the presentation. You can discern we have considerable passion on the subject and considerable experience in doing this for a number of different sponsors. And so, Sherilyn, I thank you for your time. I thank everyone for your attention. We will speak soon.
Thank you for listening to our webinar. If you have any questions or would like to learn more about how worldwide clinical trials can support your rare disease trial, please contact us at worldwide.com.